Okay, today I'm going to be talking about the topic of sensation and perception. So, first of all, it's important to distinguish the fact that sensation and perception are not the same things. Although colloquially we might think of sensation and perception as being very similar, from a psychological point of view, they're actually quite different processes. Sensation is the process of detecting, converting, and transmitting raw sensory information from the external and or internal environment to the brain. Sensation is a process that starts out by detecting a physical stimulus, and it occurs at the level of sensory receptors. Perception, on the other hand, is the process of selecting, organizing, and interpreting sensory information so that we actually understand what the sensation is. Okay, so these are two separate processes. Perception occurs at the level of the brain, sensation occurs at the level of the receptors, and it involves the process of transmitting information from sensory receptors to the brain, where perception will take place. So usually when we think about sensation and perception, we cover 5 or so senses. We actually have many more than 5 senses, but we usually talk about 5 distinct senses, and these include vision, audition, which is hearing, olfaction, which is our sense of smell, gustation, which is our sense of taste, and a variety of body senses that include things like pain, temperature, and tactile pressure senses. Now, it's important to point out at the very beginning of this that even though we have these discrete senses, they all involve very similar steps in terms of the process of going from detecting a sensory stimulus, which is sensation, to the process of perception. So if we look at these different senses in the table here, we see that each sense has its own unique stimulus. Okay, but there's a physical stimulus for each type of sense. Okay, that sense gets detected by different receptors, so there are different receptors for each sense. But once these receptors become activated, the process of sending that information from the receptor to the brain is actually the same for just about all senses. Okay, so it's really just a function of where that information is sent to in the brain that really determines what the perception of the stimulus is. Okay, so just keep that in mind as we talk about these different senses, that even though we have these different types of receptors and they go to different locations in the brain, the process is really all the same. Okay, a stimulus, a physical stimulus in the environment, it could be internal or external. Okay, because we could have pain inside of our body that's detected, or it could be external, like a light, for instance. But the guiding principle here is that there's a physical stimulus, it activates a sensory receptor, and that sensory receptor translates that information into a neural impulse, sends it on to the brain. Okay, so just keep that in mind, that that's the same process for all the different senses. Okay, so I mentioned that we have to convert this raw physical stimulus into something that the brain can actually understand, and that's a process that's called transduction. Okay, this is when again a physical stimulus that stimulates a sensory receptor is converted into a neural impulse that can be sent on to the brain. So we've talked about neural impulses. These are basically just the action potentials that travel along axons. Okay, they get from the cell body, or in this case a sensory receptor, to other neurons. Okay, coding is the converting of these sensory inputs into different sensations. Okay, so as I mentioned, we have different sensory receptors, but they all sort of travel along different pathways, and it's their final destination in this pathway, the location in the brain that the information gets sent to, essentially, that is what determines what that sensation is perceived as. Remember that sensation and perception are two distinct processes. And there's also a phenomenon called sensor reduction that allows us to filter and analyze incoming sensations before sending these neural messages on to the cortex. And this has to do with the fact that we're being constantly bombarded with a variety of sensory stimulation from our environment, and it would be very overwhelming if we were actually processing all this information at one time. So sensory reduction is a way of sort of monitoring the amount of sensory information that is being sent down these pathways. Okay, so as I mentioned, ultimately the process of perception is a process that occurs in the brain, and we have different parts of the brain that seem to be more responsible for the perception of some senses than others. Okay, and for that reason, discrete injuries to certain areas of the brain can result in a loss of the ability to detect certain types of sensory information. For instance, if you had an injury to the occipital lobe, okay, we've talked about how that processes visual information in the past. The type of behavioral symptom that you're going to see when there's damage to that area is going to involve some sort of disruption in visual processing. On the other hand, if there's damage to the somatosensory cortex in the parietal lobe, then you're going to be more likely to see a disruption in the ability to perceive the sense of touch. Likewise, if you have damage to an area called the primary auditory cortex in the temporal lobe, you're most likely going to have a disruption in the perception of auditory information, okay, and so on. So here we're just looking at the different areas of the brain that are primarily involved in the processing of discrete types of sensory information. Okay, and this is where perception will begin to take place. Okay, so in terms of the amount of stimuli, the intensity of a stimulus that is necessary to actually be recognized, because sometimes there are sounds that are too faint to be heard. Okay, that means that they're below our sensory perception, or something flashes so quickly that we couldn't quite make out what it was. Or we might fail to detect it altogether. And so an area of psychology that studies the processes that are involved in detecting these stimuli is called psychophysics. Psycho meaning the psychological experience, okay, the mental processes involved and whatnot. Physics referring to the fact that these are processes that are instigated through some sort of physical stimulus. Okay, so psychophysics studies the link between the physical characteristics of stimuli and our sensory experience of them. So, what that suggests is that there's not a one-to-one correspondence between sensory information in our environment, sensory stimuli in our environment rather, and our perception of them. Okay, a good example of this, as a matter of fact, has to do with the fact that it's very difficult to tickle ourselves. Right? We could be tickled by other people, but we have difficulty tickling ourselves. Okay, it might be the exact same sensory stimulation that causes the perception of tickle, but in one case, when somebody else is doing it, we perceive that sensation, and in the other case, we don't. So again, there's not always a one-to-one correspondence between particular physical stimuli and our sensory experience of them. Okay, so there's a concept called absolute threshold, which refers to the minimum amount of a stimulus that must be present before an observer can reliably detect it. And when we say reliably detect it, we mean greater than 50% of the time. So that is, many people in their lives have gotten hearing tests to determine what range of frequencies they can actually hear. What range of frequencies, as well as the loudness, which we'll be talking about, that is necessary to detect that sound as well. And so when you're getting a hearing test, you start out at the audiologist or whoever is administering the test will start out at an intensity that is believed to be so low that you can't hear it, and then it'll be gradually increased. And at some point, you'll begin to hear it, and before that point, you won't hear it. That is the absolute threshold. And there are some examples here of what the sensory thresholds are for some of the different senses. Okay, a different concept, but related, is called the difference threshold, or the just noticeable difference. This refers to the minimal difference needed to notice a stimulus change. So here the question is not, is a stimulus present or absent? It's a question of, are these two stimuli of equal intensity? So for instance, if you hear one noise and then you hear, if you hear one sound, and then another sound, are they the same sounds or are they different? And how different would they need to be before you could begin to perceive that difference? That's called the just noticeable difference. And just a quick note on the topic of subliminal perception, because there's quite a few people out there that believe that we can be persuaded and influenced by stimuli that are, again, below this sensory threshold, so that we can't consciously determine what it is we saw, but we can still be influenced by it. That's what subliminal means, below the limen, which refers to this limit of intensity that we would need to be above in order to detect something. So subliminal perception is perceiving something without a conscious awareness. So in other words, we don't know what we saw, yet there are certain indicators that can be demonstrated that strongly suggest that we did in fact detect something. We just don't know what it was. So I just want to mention at the outset that there's an extremely small likelihood that subliminal perception can have a significant influence on us. It's not to say that we can't notice very subtle changes in our behavior, but it's not as if you're going to hear a subliminal advertisement on the television and then you're going to go out and buy a product because of that. Okay, it's a much more subtle effect than that. Now, another thing that we see when it comes to sensation is a process called sensory adaptation. Okay, this is the gradual weakening of a response that's due to repeated or constant stimulation. So, in other words, the longer we're exposed to something, the less sensory stimulation it evokes. So this is why you put on cologne or perfume in the morning, and then a few minutes later you don't notice that you're even wearing it anymore, yet you go to see your friend and they say, oh, I love the smell of that perfume, which you had just adapted to, and you were not even aware of that smell anymore. So sensory adaptation is a good thing because it allows us to, again, not be bombarded by too much overload of sensory stimulation and information. Now, we see some similar processes when it comes to pain perception. Okay, the predominant theory on the way that we perceive pain, because the way that we perceive pain is influenced by a number of factors, not just the painful stimulus itself. One theory that attempts to explain why it is that we can experience different amounts of pain with different stimuli, or different amounts of pain with the same stimuli, is that pain sensations are actually processed and altered by mechanisms within the spinal cord. So, for instance, when there's tissue damage, which is the main reason why we experience pain in the first place, when there's tissue damage, this opens a gate, and that allows the pain signals to pass through it. Counterpressure actually has been shown to close the gate, and this is kind of the reasoning behind why, if you rub a painful area, that can actually mildly reduce the amount of pain that you experience. But as I said, pain is really a complex mix of both biological and psychological factors. Okay, perfect example of this is a football player who has an injury during the game but continues to play through it. Anybody who's ever played a sport or engaged in some sort of athletic activity knows that when you're in the middle of the athletic event, you just don't perceive the pain the same way. You can sort of run through it. part of the reason that this might occur is because we produce a chemical substance ourselves called endorphins. Okay, we touched upon these in an earlier lecture. These are neurotransmitters that act very similar to the way that morphine acts by, again, inhibiting pain perception and closing the gate. Endorphin actually means endogenous morphine. That's where that word comes from. And the main chemical that gets produced when there is this tissue damage is called substance P. P standing for pain. This is the chemical that sort of opens that pain gate and appears to be the main chemical responsible for the experience of pain. Okay, so first off, before we start talking specifically about vision, it's important to point out that the properties of vision and hearing share some important similarities. That is, the way that we perceive visual information and the way that we perceive auditory information is based on the transmission in the physical environment of sound waves, okay, sound waves in the case of sound, but light waves in the case of vision. So a couple things that we need to be aware of is that there are different properties of these wavelengths. Okay, wavelength could vary in terms of the length between the waves. This is wavelength. This is the distance between successive peaks. Okay, so when you have long wavelengths, these correspond to they're low frequency because wavelength is measured as the amount of oscillations over time. Okay, and that's what frequency is. They produce the perception of reddish colors and low-pitched sounds. Okay, so when you have low wavelengths, that's perceived as low pitch in terms of sound waves, and reddish colors in terms of wavelengths of light. On the other hand, short wavelengths, which are high frequency, are perceived as bluish colors in terms of light, and high pitched sounds in terms of sound waves. Okay, the other property is the wave amplitude. This is the height from peak to trough. So low amplitude or low intensity corresponds to either dull colors or soft sounds, whereas high amplitude or high intensity corresponds to bright colors or loud sounds. And then finally, we can also have a range of different wavelengths present at the same time. Okay, and this is what gives sound its complexity, either more complex or less complex, as well as the complexity of colors as well. Okay, so in terms of this visual processing, okay, we start out with light entering into our eye, and that information, again, there's going to be a similarity here. A physical stimulus is detected by some sensory receptors. The sensory receptors translate that physical stimulus, the light in this case, into a neural impulse, and then it travels along a pathway to go to the brain. Okay, so we'll look at this in more detail in just a minute, but this is what I was talking about a second ago with the different wavelengths of light. So, as I said, longer wavelengths are perceived as reddish colors, whereas shorter wavelengths, higher frequency wavelengths, on the other hand, which means that there are more cycles per second, are perceived as blue. Longer wavelengths are perceived as red. Okay, they're longer wavelengths because there are fewer of them in a unit of time. And again, that's how we measure frequency, is the number of oscillations in this case in a unit of time. Okay, so this is light is really a form of electromagnetic energy, but radar, microwaves, X-rays, and gamma rays, they're also forms of electromagnetic energy, but we don't have the sensory receptors to detect those things, and so they're essentially invisible to us. Okay, us meaning humans, but other species are capable of detecting ultraviolet rays of electromagnetic radiation. So these various types of electromagnetic waves, they form the electromagnetic spectrum. Okay, so but we're going to be focusing on just this this range that we're capable of detecting, and we call that the visible range of light. Okay, so as I said, light starts off by entering into our eye. It passes through a structure called the cornea first. Okay, that's the outermost portion of the eye. Then it goes through the pupil. That's the black part of the eye. It constricts and dilates based upon the constriction and relaxation of some muscles surrounding the pupil, called the iris. That's also what gives your eye its color. Okay, it passes through an aqueous solution called the vitreous humor, and it's detected by sensory receptors at the back of the eye. Okay, so light that is in our visual information that is in our that is most clearly seen is most clearly seen because it's the visual information is entering our eye and going to a specific part of the back of the eye, which is the retina, called the fovea. Okay, this is a tiny pit in the center of the retina that's filled with a type of visual receptor called cones. Okay, and as I said, this is responsible for sharp vision. So, if you're just looking ahead right now, whatever is most clear to you that you're looking at is going to your fovea. Now, you can see things off to the left, to the right, bottom, and top of your visual field, but they're not quite as clear. So those things that are not quite as clear are not going directly to your fovea. Okay, only the thing that is most clear in your view right now is being transmitted directly to your fovea. And the reason that it is the most clear is because it has the highest density of these visual receptors that we call cones. Okay, there's another type that we're going to talk about in a minute. There's also you'll also notice that you can see a blind spot in your visual field. Everybody has it. It's perfectly normal. You can only see it when you have only one eye open at a time because you have a different blind spot in both of your eyes. But this is the point at which the optic nerve, okay, this is the bundle of axons that leaves the eye and goes to the visual cortex in the brain before making some stops at various other locations first. It contains no sensory receptors, and that's why we don't see anything there. Okay. Another important process to be aware of is called accommodation. This is the automatic adjustment of the eye, which occurs when muscles change the shape of the lens so that it focuses light on the retina from objects at different distances. So again, if you're looking straight ahead, you could look at something that is close to you or something that's far off in the distance without really changing the direction in which you're looking at, and that's because of accommodation. Okay, so here we're looking at a more close-up of the retina. The retina is, again, this is a light-sensitive area on the inner surface of the back of the eye, which contains the receptor cells for vision, which are called rods and cones. Okay, cones we just talked about a minute ago. These are visual receptor cells that are concentrated near the center of the retina. Mostly, they're most highly concentrated in that area called the fovea. Okay, they're responsible for color vision and fine detail. Okay, they are most sensitive in brightly lit conditions. In other words, it takes a lot of light in order to activate them, and hence that's why they're better at perceiving fine detail, because they function optimally when there's a large amount of light present. Okay, in a dark room, you can't see very well. That's because largely your cones are not being activated. You can see though, and that's because rods, the other type of visual receptor cell, they're best for detecting shades of gray and for peripheral vision. Okay, they're most important in dim light and at night because they're more sensitive to light, which means that they require less light to be present in order to be activated. Okay, so to sort of summarize up what we've been talking about, light first enters through the cornea, which helps protect the eye and focus the incoming light rays. Okay, this is the area that typically gets changed during eye surgery to help with the direction of that light. Light then passes through the pupil, which is the adjustable opening in the eye controlled by the iris, which is the muscle. Okay, in the retina, light waves are then detected and transduced or converted into neural impulses by these receptor cells called rods and cones. Remember that the area where we have the highest concentration of cones, the area that is responsible for our most clear vision, this is the fovea. Okay. From the sensory receptors, okay, that information gets passed on to the axons that make up the optic nerve. Okay, from the optic nerve, they will travel to the visual cortex. Okay, so again, this is something that is often used to demonstrate this blind spot. Again, you have to close one of your eyes. You stare at the X and you move your head slowly toward the screen, and at some point, there's a portion of the apple where the worm is that will actually disappear. So there's a number of instances that you can look at this blind spot from. Okay, so moving on to audition. Okay, our sense of hearing. We're going to see a very similar process. Okay, we're talking about waves again, sound waves this time instead of light waves. Sound waves enter the ear. They do a couple of things first. Move some structures within the middle of the ear, but the movement of those structures in the middle of the ear causes movement of another portion inside the inner ear that stimulates sensory receptors. These sensory receptors in the ear are called hair cells. The stimulation of these cells triggers neural impulses to be sent along the auditory nerve. Okay, so again, just want you to keep in mind that similarity in terms of these are different types of physical energy, but they're all being detected by different types of receptors and then transducing that information into a neural impulse. And so the process is actually somewhat similar across these discrete senses. So a couple of things to take note of in this diagram here. We've got the ear is typically divided up into three discrete units. The outer ear, which is largely the area that we can see, although it also includes an area called the auditory canal, which is the part that goes inside of our head. The middle ear and the inner ear. Okay, so the outer ear captures and funnels sound waves into the inner ear, bypassing through the middle ear first. Okay, vibrations of the tympanic membrane strike the middle ear's ossicles. Okay, these are the three smallest bones of the body. They're referred to often as the hamel, anvil, and stirrup, more technically known as the malleus, incus, and stapes. And they actually put pressure on another membrane called the oval window. The vibration of the oval window creates waves in the inner ear's cochlear fluid, which deflects the basilar membrane. Okay, so in other words, inside this structure that looks like a snail shell called the cochlea, there's a fluid inside of it, and when you have sound waves putting pressure on the membrane, that pushes that fluid. When you get movement of the fluid, it causes the membrane that is surrounding that fluid to move. Okay, when you move that membrane, that's when you start to move these cells that are called hair cells, and they literally look like hair cells. That's why they were named like that. So you can imagine, just gently grazing your hand over the top of your head, and you can feel that there's going to be some movement of hair cells. If you have short hair like I do, it's probably a good representation of it. You just softly brush your hand along your head there, and you're going to move those hairs, and picture the movement of those hairs is actually generating action potentials. Okay, this is what's going on in the inner ear, but it's also what's allowing you to feel the movement of your hair itself. Okay, it's a different process, but it's analogous to what's occurring in the inner ear. Okay, now there's a couple different theories about how we actually distinguish different sounds of different pitches. Remember that pitch is determined by the frequency of the sound waves. That is, how many times it oscillates in a second, which is referred to as hertz. Okay, so one theory called place theory explains pitch perception as being linked to a particular spot on the cochlea's basilar membrane that's being stimulated. So, in other words, high frequency sounds stimulate areas further down along the cochlea, along the basilar membrane, whereas lower frequency sounds stimulate areas that are located not as far down on the basilar membrane. In other words, closer to the beginning of the inner ear. Frequency theory, on the other hand, explains that pitch perception occurs when nerve impulses are sent to the brain that match the frequency of the sound wave. So, in other words, low frequency sound waves will cause the basilar membrane, and therefore the firing of the hair cells, to occur at a slower rate. Okay, so examples of low frequency sounds that are perceived as low frequency sound waves that are perceived as low pitched sounds would be, a man's voice, versus high frequency sounds. An example would be a child's voice. Now remember that frequency is independent of loudness. Okay, loudness is based upon the amplitude of the sound waves, and it's measured in decibels. So just to give you an idea of what kinds of different noises are associated with what decibels, you can look at this figure here, and you can see that anything above 90 decibels can result in nerve damage. So that's, of course, why you don't want to listen to your iPod or your music at loud volumes, specifically volumes that are over 90 decibels, because prolonged exposure to that can result in permanent damage. Okay, so the next sense that we're going to talk about is olfaction. Okay, this is a chemical sense along with our sense of taste, gustation. Those are the two chemical senses. This is our sense of smell. Okay, so what happens here is we have chemicals. Okay, airborne molecules are detected by these olfactory receptors. Okay, these chemical molecules are just referred to as odorants. They get they attach to these olfactory receptors that stimulates action potentials. They go on to the area of our brains called the primary olfactory cortex. In addition to the thousands of different types of smells that we're capable of distinguishing between, there's a specific type of chemical that's actually a little bit controversial in terms of the degree to which it actually applies to humans. It does appear to apply to humans to some extent, but not quite as much in humans as in non-human animals. Pheromones are compounds that are found in the body sense that have been shown to impact various behaviors, and largely these are related to sexual behaviors. So just to give you a quick example of this, there was a study that was done where they looked at, they had undergraduate students who were research participants, volunteered for a research study. They had them smell T-shirts that had been worn by various people overnight who didn't wear deodorant or eat any spicy foods or garlic or anything like that. They had these research participants smell the different shirts and rate them in terms of how pleasant or unpleasant they were. Now, I don't think that any of them were rated as particularly pleasant, but some were rated as more unpleasant than others. And it turned out that the individuals who were rated as being less unpleasant, that is more pleasant, were the same individuals who were rated as having more attractive faces. So there seemed to be a correlation between facial attractiveness and the pleasantness of their pheromones. Okay, again, the idea here is that these pheromones are located in our body odors. Okay, that's a relatively subtle effect, but it's much more prominent in non-human animals, to the point where the presence of pheromones can actually induce, in some some species, can actually induce females to begin ovulating. Okay, so I mentioned that the two chemical senses, olfaction and gustation. Gustation refers to our sense of taste, which occurs primarily on the basis of chemical molecules stimulating sensory receptors called gustatory cells. These are taste receptors. But again, the process is all the same though. Travels, stimulates these receptors in our tongue, travels along nerve impulses, goes to an area that we call primary gustatory cortex. Okay, the five different discrete taste sensations that we seem to be capable of discriminating between are sweet, sour, salty, bitter, and umami. So if we think about all that's involved in our experience of eating a slice of pizza, okay, there are, we can see each of the different pathways being activated here, right? We've got the visual pathway, which is what the pizza looks like. We've got the olfactory pathway, which is what the pizza smells like. We have the taste pathway, which becomes activated when we start ingesting the pizza. But we also have the tactile or somatosensory pathway, which is the texture of the pizza. So, for instance, is it hard or soft and gooey, or the texture of the pizza itself. So all these things become simultaneously activated. Okay, so I mentioned that we do have more than five senses, and a good illustration of this is the fact that we have different types specifically of body senses. So we have skin senses, vestibular senses, and a sense of kinesthesis. So the skin senses, these are tactile senses that rely on a variety of receptors located in different parts of the skin, but they perceive things like pain, temperature, and pressure. Pressure is largely responsible for us being able to perceive the sense of touch. Okay, and we actually have different types of receptors for these different skin senses, and they can become even more complex. For instance, I mentioned tickling before. Tickling and itching are examples of combinations of senses. The sensations of itching and tickling are actually a result of the simultaneous stimulation of both pain and pressure receptors. So our vestibular senses, these are responsible for our sense of body movement and position, also called the sense of balance. Okay, our sense of our vestibular sense is a result of structures that are located in the inner ear. Okay, so if you actually look at the diagram of audition, we can see that there are these additional structures that are located right near the cochlea called semicircular canals. Okay, and inside those semicircular canals, we also have structures called vestibular sacs. So the vestibular sense, as I mentioned, is responsible for balance. It informs the brain of how the body, and particularly the head, is oriented with respect to gravity and three-dimensional space. So, for instance, when the head moves, the liquid in these semicircular canals will move, and when that liquid moves, just like we talked about before with audition, that causes hair cell receptors to bend, triggering action potentials. Okay, and that's what informs our brain of the fact that we're our head is looking downward, for instance, or that we just experienced. We just turned our heads very quickly. At the end of these semicircular canals, as I mentioned, are vestibular sacs. These also contain hair cells that are specific to the angle of the head, whether it's straight up and down or tilted. So, both of these senses help us to identify the direction and location and movement of our head. Kinesthesis, on the other hand, is a system for detecting body posture, orientation, and movement of individual body parts. So, for instance, if you close your eyes and raise your hand, you know that your head, you know that your hand is located above your head. And the reason that you know that is because there are these receptors that are found in muscles and joints and tendons. And so, for instance, when you go to contract your bicep muscle, that causes the stretching of your tricep muscle, and that's one of the things that informs you that your arm is bent. On the other hand, if you contract your tricep muscle and relax your bicep muscle, that causes your bicep muscle to stretch. It also causes the stretching of these receptors inside of the muscles, and that tells you that your arm is straight. And so that's largely what makes up your kinesthetic sense. And again, these receptors are also located in joints and tendons as well.